Yeah, so what we did is we looked at the validation of MRD as a surrogate endpoint in AML. And that's a lot of difficult words, I guess, for someone that's not in the field. But what it comes down to is that we see that for new AML drugs, it usually takes a lot of years to evaluate if it's beneficial for the patient. And that's mainly because the endpoint relies on overall survival. And this can only be measured after 3-4 years of follow-up. In other blood cancers, so for instance, multiple myeloma, an earlier marker called measurable residual disease, or MRD for short, is already used to predict outcome and to guide the drug approval. So it's used as what we call a surrogate endpoint. And what we wanted to test in this study is to see if MRD could also play the same role for AML, which would allow us to identify that effective treatment much earlier than we can do now. The results in the study provide a quantitative evidence that MRD is strongly linked to patient survival. And we can see that if the treatment improves the overall survival, then it also tends to improve MRD response. And that supports using MRD as an intermediate endpoint, which is so a tool for regulators to use for accelerated drug approval. And that's mainly as an sort of intermediate endpoint, meaning that a drug can already be approved while we wait for longer term data on overall survival to mature. Yeah, so multiple myeloma is more or less our thing we looked at the most, mainly because the FDA recognized recognized the multiple myeloma MRD as an endpoint. They did that with a what we call moderate trial level correlation, so an R squared of around 0.7. And the correlation we could find in our AML study was stronger than that. But there was some difference between patients that were transplanted or not, because the correlation was the strongest in patients that did not go to transplant afterwards. So I would say this study is making a convincing example that MRD can or has a predictive power in AML. Yeah, that's a good question, and I think overall survival will remain and will always remain the ultimate measure of success. It's easy to assess, and yeah, it's in the end the most important thing. But the problem is that it takes years to reach in AML. Think about 4 or 5 years at least. So validating MRD as a surrogate endpoint could mean that we can evaluate new therapies within months instead of years. So it will help the patients mainly to access effective treatments much sooner than we can do now. But of course, the confirming benefit needs to be done later through survival follow-up. It all comes down, of course, to approval from the regulatory body, so the FDA in the US and the EMA in Europe. But if MRD is accepted as an intermediate endpoint, it could be promising for new drugs to receive a provisional approval based on the MRD, on early MRD results. And then the trials would still continue in a sort of two-stage approach, which could then shorten the timeline and trial size, but still needs the overall survival to approve for or to be assessed for final approval. So I think the most striking result that we found in the study is that patients who cleared MRD after treatment were more than twice as likely to be alive at follow-up compared to patients who still had MRD detected. And of course, this has already been shown in other studies and even in meta-analysis. But within these studies, we were never able to correct for other variables because that would mean that you need patient-level data. And in this analysis, we were able to do this to show that the prognostic value of MRD persists, but also that it's even stronger than most of the mutations that we found, for instance. So in simple terms, when no leukemia cells can be found using these very sensitive tests, the chance of long-term survival improves dramatically. Of course, the first priority is to finish this project. We have already a few years ago and recently, again, we tried to identify more studies that were conducted in Europe to enhance our results. Unfortunately, not a lot of studies in the near future will come available that meet the inclusion criteria. So we will continue with the studies that we have. And another thing that we are thinking about is trying to expand the analysis to less intensive treated AML patients, for instance, on venetoclax-based therapy. But this will probably be something not for the near future, as there's not a lot of data at this point to do these kind of analysis. Yeah, I think this study was only possible within Harmony. Harmony really allowed us to combine standardized patient-level data from several large cooperative groups in a secure environment. So that, yeah, without the platform, we would have differences in data formats, we would have variable definitions, and yeah, the privacy rules would have made this type of large-scale analysis nearly impossible. So the whole harmonization process ensured that we have results that are directly comparable across the trials. But also having this secure environment means that all the data providers can trust that there won't be any like additional or strange things be done with their data. Yeah, so the Harmony framework provides both the legal structure and the technical infrastructure for data sharing. So again, it creates a trust between the groups, streamlined ethics, and data protection in general. It will allow the statistician to do these kind of pooled analysis without compromising on patient privacy. So yeah, I think it instead of having national efforts, it now turned into coordinated international projects within Europe. Yeah, so I cannot stress this enough, but these kind of analysis are only possible with large data providers coming together and trusting each other to do these kind of things. No single trial could have ever resolved this question on its own. So we need groups to collaborate, and in the end, I think everybody wins. We gain statistical power, we shorten research timelines, and it generates evidence that we can use in clinical practice and for regulatory policy, which we are just not able to do as a single group. So we need cooperative data sharing in general, I think in hematology, but in all fields, I would say. unfortunately, not everybody was able to share their data, and that's just really unfortunate because the data is so important for us to move this field forward. And if people have the data locked in somewhere and cannot be used for any other purpose, then it's just sort of a waste of all the effort that's been put into the data. So we really need all the data that we can have, and hopefully that will people will put like the patient first, and the patient will only benefit if we can work together.